Louping is a tick-borne neurological disease that mainly affects sheep and grouse, and which is responsible for serious losses for hill and moorland livestock farmers and gamekeepers. The clinical signs in sheep include fever and depression, which lead to muscle tremors and difficulty in walking, and eventually to paralysis and death. Louping has been studied for many years at Moredun, and our scientists have been instrumental in understanding the cause of the disease and in developing vaccines to protect the UK's sheepflock. Modern scientists first identified a tick-borne virus as a cause of louping in the 1930s. They then very quickly developed the first vaccine against the disease. This first vaccine was a killed vaccine prepared from the tissues of infected sheep, and although rather crude, it was used successfully until the 1960s. By that time, it was clear that this basic vaccine could no longer be produced due to safety concerns. And the next vaccine Moredun developed was grown in cell culture and used from the early 1970s until 2017, when it was withdrawn from the market for commercial reasons. The withdrawal of the louping vaccine left a challenge for farmers and gamekeepers trying to control louping, and it was clear that a new vaccine was needed. So Moredun began working on the development of a new type of vaccine for louping. Both of the previous louping vaccines required preparations of the whole virus that were killed by chemical treatment, but in a way that retained the ability to stimulate an immune response. In the time since those vaccines were developed, a lot has been learned about the biology of louping virus and other related viruses, and we now know that much of the immune response is directed at proteins on the surface of the viral particle. At Moredun, we've developed a method to produce those proteins in the laboratory, which is simpler and much safer, as there's no need to use a live virus to make the vaccine. To formulate the vaccine, the viral proteins are mixed with other immune-stimulating compounds, and once administered to sheep, the immune system recognizes the viral proteins and produces antibodies. These antibodies remain in the body for a time, ready to respond if the virus is detected, before it has a chance to cause serious illness. The vaccine produced by Moredun has been shown to be highly effective in research trials, but now needs further testing before it can be produced commercially. You can learn more about this and our fundraising campaign on our website.